All right, so we've looked at two different types of mechanisms, those being acid-base catalyzed reactions and metal ion catalyzed reactions. Now we're going to move on to covalent catalysis. For covalent catalysis, we're going to look at the enzyme lysozyme. Now, lysozyme is an enzyme that hydrolyzes the peptidoglycan layer of bacterial cell walls. So if you've taken a microbiology class, you've maybe talked about lysozyme because, well, it helps break down bacterial cell walls. Excuse me. Lysozyme is a very regularly used enzyme in biochemical research. For this reason, you can break down biological or you can break down bacterial cell walls and then be left with the cells remaining pretty easily. Specifically, lysozyme breaks a beta 1,4 glycosidic linkage between two individual monomeric units from the sugar. So our substrate or our target is this molecule that has repeated elements of NAG, NAM, NAG, NAM. That NAG is N-acetylglucosamine and NAM is N-acetamuramate. M-acetamuramate. So we have these individual aminated sugars repeating over and over again. It's important that you recognize it is a beta glycosidic linkage. Now, what we're going to have, or what we're going to result from this, is we are going to simply have this bond right here broken, and NAM and NAG, where there previously was a linkage, there is no longer. And this is something that lysozyme will do somewhat repeatedly. And you might look at this molecule and say, oh, so it's just going to break it down into a bunch of dimers of NAG and NAM. And it's not quite that direct. And I hope that you'll see why in just a moment. So one of the things that's very important for lysozyme to be functional is positioning of this repeated sugar, this repeated sugar structure kind of within its enzyme active site so that it can be broken. Now, if you look over here on the right, what you see are NAG, NAM, NAG, NAM, NAG, NAM. We have a total of 6 different sugars within this oligosaccharide. Now, or that are shown here in this oligosaccharide. Now, what I want you to first recognize is lysozyme is going to cut between what is the 4th and 5th sugar in this oligosaccharide. The the NAM and NAG, it's going to cut there. So this location right here that we were referencing is the exact same as this location right here. Now, the fundamental difference is obviously the positioning of things. But one of the things that I want to draw your attention to, and that I think is pretty cool, is we've got all these different amino acids. Sorry for yawning. We've got all these different amino acids. Tryptophan 62, tryptophan 63. Up top, we have aspartate 101, alanine 107, glutamine, asparagine, phenylalanine, asparagine, arginine, all of these different amino acids that are helping position this oligosaccharide in the correct location so that we can actually break this glycosidic linkage. Now, what I want you to recognize about this is really let's see, I'm going to count that off. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, maybe 16. A quick count, there's 16 different amino acids that are identified as establishing a bond between our substrate and the enzyme itself. Now, all told, the amino acids that are responsible for breaking the bond, breaking this beta 1,6 1,4 glycosidic linkage, sorry, 1,4 glycosidic linkage, it's really down to about 3 different amino acids. And I'll show you 2, maybe 3 amino acids, and I'll show you that in the next slide. But I do want to draw your attention to a handful of these different amino acids that are kind of, well, they're not so much, what they're doing is basically helping position the bond in the right location so that the amino acids that break that bond are right there to do that. Now, what I'd like to draw your attention to first is this aspartate 101. By the looks of it, from where our lysozyme is actually going to cut, that amino acid might be the farthest from that cut site. But what's it doing? Well, what aspartate 101 is doing is it's establishing hydrogen bonds with parts of this sugar, parts of this oligosaccharide to more or less hold it in place. Now we know that hydrogen bonds aren't particularly strong, but when you have lots of them, they're quite a little bit stronger, or they're quite a bit stronger. Now aspartate 101, that's a polar negatively charged amino acid. So it can participate in ionic bonding or hydrogen bonding. So the fact that it's establishing a hydrogen bond isn't really a stretch of the imagination. Next up, if we look at tryptophan. Oh, tryptophan, that's interesting. Tryptophan is establishing a hydrogen bond with our oligosaccharide. Man, tryptophan's a relatively non-polar amino acid. It's one of our aromatic amino acids, but it's able to establish a hydrogen bond. We've got tryptophan 62 and 63, which are both contributing hydrogen bonds to help stabilize and position this oligosaccharide so it can be broken down. Next up, sorry, an amino acid that I think is really kind of the neatest, alanine and phenylalanine are both examples of amino acids that, well, when you think about catalysis, when you think about the R groups, well, alanine and phenylalanine, at best, those are extremely nonpolar. These two amino acids really don't like to share any sort of hydrogen bonds. You know, those are more than likely going to be found in hydrophobic interactions. But what I want to draw your attention to is what's the group that, that we have we have a bond between the letters in black indicating our alanine 107 and the letters in kind of a rust or maroon color indicating part of our oligosaccharide. Well, those are backbone interactions. So the backbone of this polypeptide, specifically at an alanine 107 and phenylalanine 304, they are participating with stabilizing the structure so that the molecule can be broken, or so that the oligosaccharide can be cleaved. Now, what we're seeing happen in this reaction is actually, if you remember the cyclization of a sugar, we went from an aldehyde to a hemiacetal, and then a hemiacetal would react with an alcohol to form an acetal. Well, in this case, we're kind of going in the opposite direction. We're taking an acetal, which is our oligosaccharide, and we're going back to a hemiacetal. So we're still ending up with a molecule that has a cyclic sugar on it. This is just kind of reversing the reactions that we previously talked about. Okay, now here it is. This is our catalytic mechanism that we use to study the enzyme lysozyme, or this is the catalytic mechanism that's been presented to best describe the reaction catalyzed by lysozyme. Now, what I'd like to draw your attention to are our two amino acids, glutamic acid 35 and aspartic acid 52. Now let's jump back for a second. Do we see those? Well, here's by where lysozyme cuts. there's aspartate 52 and glutamic acid 35. So those two amino acids arguably are our two amino acids that are most central to catalyzing this reaction. Now, this is a covalent mechanism, or this is a covalent catalyzed reaction. So what we should expect to see is a covalent bond between our enzyme and our substrate. So either glutamic acid 35 or aspartate 52 is going to establish a covalent bond with our substrate. Now, let's look at that glutamic acid 35. That glutamic acid 35, I'm going to start in the upper left-hand corner. And well, it is protonated. It is protonated, therefore I'm using the correct terminology of glutamic acid 35, not glutamate 35. Glutamic acid 35. What it's going to do is it's going to offer up that hydrogen ion right here in order to break this bond. So in order to break our glycosidic linkage, we're going to offer up that proton. So GLU35, what it is doing is it's acting as an acid. Okay, that's worthwhile to note. So if this somehow were the end of the mechanism, well, it really wouldn't be covalent catalysis because we didn't see a covalent bond formed between our enzyme and our substrate. We saw our enzyme donate a proton. So let's go ahead and take a closer look. We have aspartate 52. Aspartate 52 is deprotonated, which is why I'm saying aspartate and not aspartic acid. So aspartate 52 is deprotonated. And whenever glutamic acid 35 donates a proton in this first step, moving from cell number 1 to cell number 2, well, aspartate 52 doesn't do anything. Instead, it just observes that the covalent linkage, the beta-1,4 glycosidic linkage is broken. Our substrate finds this transition state, this oxonium ion. And in this oxonium ion, we have a negatively charged group for that aspartate 52. So glutamic acid, or glutamate 35, is present in this second cell. Aspartate 52 is also negatively charged. And what aspartate 52 does is it goes ahead and acts as a nucleophile, and it's going to make the covalent bond between itself and our substrate. So we're going to stabilize our substrate by eliminating that oxonium ion. And here it is. In our third cell, we have that covalent linkage. We have the covalent linkage between aspartate 52 and our substrate. Hence, this mechanism would be identified as involving covalent catalysis. Okay. Now, with that said, our entire reason for studying enzyme mechanisms is in benefit to studying biological catalysts, as we have a start point and our end point that are identical. So we need to get the glutamic acid 35 back to its initial state and aspartate 52 back to its initial state. At the moment, we have glutamate 35 and we have, well, aspartate 52 with a covalent bond to this sugar or this oligosaccharide. So now what we're going to do is we're going to introduce water. What water is going to do is it is going to act as a acid, and it's going to act as a base. And this hydroxide ion and the hydrogen ion that make up water, well, we're going to be able to protonate glutamate 35 to make glutamic acid 35, which is where we are in this top cell. Then what we're also going to be able to do is the hydroxide is going to be able to break the covalent linkage between aspartate 52 and our substrate, which will enable our substrate to leave And we've gotten back to where our enzymes were at the beginning. Glutamic acid 35, aspartate 52. So we're in good shape there. There's a lot there's a lot to unpack from this. Was there acid-base catalysis? Yes, there was. Acid-base catalysis was a key part of this. Was there a covalent catalysis? There absolutely was. Now, this is classified as covalent catalysis because a covalent linkage is established. This is not classified as being like totally acid-base catalysis because this acid-base catalysis differs quite significantly from our RNase A mechanism. In our RNase A mechanism, did we have a covalent bond between our enzyme and our substrate? No, we did not. But here we do, which is why this is classified differently. I hope that was helpful. I recommend anytime you look at a mechanism to move through it slowly and deliberately, to look at that first cell and migrate through very, very slowly and carefully. What is each amino acid doing in any different position? And here we're focused on two of our acidic residues, glutamic acid and aspartic acid. Do they do the exact same thing? No, they don't. One is an acid or one is involved in acid-base catalysis and one is involved in covalent catalysis. So now we move on to a discussion of a class of enzymes known as serine proteases. And these are another class of enzymes where we'll look at the mechanism, but I wanted to give you a little bit more of a global perspective. Serine proteases are a family of proteolytic enzymes that have an active site serine. So this is kind of a classification exercise. These are serine proteases because within the active site, there is a serine, as opposed to, well, they look for a serine and then they cut after or before a serine. Now, a protease is a type of enzyme that breaks a peptide bond. So you could kind of think about these as proteins that cut up proteins. Now, here's two examples of serine proteases, the structures of these serine proteases. And it's not so mission critical that you can see, oh, well, that's chymotrypsin clearly because there's an alpha helix at this termini or anything like that. But instead, there's a couple of amino acids that are highlighted. And these amino acids are right here and right here, respectively. Now, what I want you to take away from this is when you look at structures like this, it's important to be able to kind of say, okay, well, I see those balls and sticks in space. Those are making up a molecule. Those are atoms, okay? Some are red, some are black, some are blue, some are black. Well, think about these different groups right here and try to visualize, okay, well, if I've got two red, like let's take this one right here. Try to decipher, try to think about your amino acid structures and think about, well, what does the structure consist of? Okay, so if I've got black, 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 and then red and red. Okay, so essentially the black ball is kind of the most common one that you see. So I think that it's not too much of a jump to say, well, that's probably carbon. In which case, let's rewrite this. Okay, so carbon, 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 structure CH2, CH2. Okay. You know, some of y'all may be very adept at looking at this and saying, I know that's this amino acid, but others you might be kind of tripping up on this. So I've got X and X, so two identical atoms Okay, so what could they be? Could they be nitrogens? Well, I didn't really see any amino acids that had a carbon and then two nitrogens coming off of them. But it's obviously not carbon, so maybe let's try oxygen, because oxygen, carbon, nitrogen, hydrogen are basically your only atoms within a molecule, in an amino acid. So does it make sense that maybe this is it? Yeah, I think so. So we've got either an aspartic or a glutamic acid, depending on if this is highlighting, like, is that carbon a part of the R group or is that part of the side chain? The important thing that I want you to take away from this is being able to look at a structure and saying, oh, I bet that's histidine. So it's possible that this is my histidine, or this is a histidine, this is a glutamic acid. Maybe that is my serine, because I'm kind of expecting to have a serine in this. Okay. With that said, there's another serine protease known as thrombin, which is a critical enzyme in blood clotting in order to initiate blood clotting. Trypsin, chymotrypsin, and elastase are all digestive enzymes, so they're found in gut or in your gut. Subtilisin is a bacterial protease. Plasmin is also a serine protease that breaks down the fibrin polymers of blood clots. So these are all serine proteases. And so within the active site, so within the catalytic core of the enzyme, there is a serine. Tissue plasminogen activator is or TPA, cleaves the proenzyme plasminogen, which produces plasmin. Now, that suffix there that you maybe saw, ogen, plasminogen, and plasmin, that terminology, plasminogen getting cleaved to make plasmin, that isn't a mistake. Remember, proteases are types or are enzymes that cut peptide bonds. So they're proteins that break down proteins. Now, there's lots of different layers of regulation of enzymes and processes in a system, in a biological system. And one more layer to that is this class of molecules known as zymogens. Zymogens are basically inactive proteases. Zymogens are proteases that get activated by proteolytic activity or proteolytic cleavage. So some enzymes are initially produced in an inactive form known as a zymogen. A portion of the protein chain must be removed to make it active. This is an irreversible action. Now, what we look at as an example is, well, there's an example shown over here to the right. That is, you have blood clotting factors. Prothrombin is a zymogen that is activated by binding of calcium, which leads it to be cleaved to make thrombin. Thrombin activates fibrinogen to make fibrin. Now, to illustrate these a little bit more, I would like you to look down at the lower left-hand corner. We have alpha chymotrypsin, or for our purposes, chymotrypsin. Chymotrypsin is an active enzyme that has basically 240 some odd amino acids. It's got a bunch of disulfide bonds that hold it together. This structure right here that's shown is really just a simplistic model. Sure, it's going to go ahead and fold into these unique structures to make that three-dimensional structure. One thing that's worth noting is this would be a quaternary structure because it's multiple polypeptides that are going to fold together. Now, this is the active version. But in order to become fully activated, a couple of things have to happen to chymotrypsin. One, whenever it gets translated, so going from DNA to RNA to a protein, we produce the protein chymotrypsinogen. Now, chymotrypsinogen and chymotrypsin these are digestive enzymes. So chymotrypsinogen is generated, and you know what? It's just going to float around. It's not going to do anything. It is inactive. It cannot break a peptide bond. However, when the time is right and when the season is right, there is an enzyme known as trypsin that comes along, and what it does is it breaks a bond right here between amino acids number 15 and 16. That makes a protease, or that makes a derivative version of chymotrypsinogen known as pi-chymotrypsin active. And whenever pi-chymotrypsin is active, what it's able to do is it is able to cut itself. And so pi-chymotrypsin can't cut anything else, but it can cut itself. And whenever it cuts itself, it goes ahead and cleaves out some dipeptides. It cleaves out a dipeptide of serine and arginine, and it cleaves out a dipeptide of threonine and asparagine. So these red regions within pi-chymotrypsin are simply cut out by itself. It's almost like, hey, I don't need these anymore, gone. So then alpha-chymotrypsin is going to fold into itself, and it's going to be able to be completely active, in which case it can go on, and it has its own selective mechanism to cut specific peptides or not cut them. But it's not until it's fully activated by the cleavage by trypsin and then by itself where it can cleave other polypeptides, or kind of polypeptides from itself that are different from itself. Now, these digestive serine proteases, they have another structural element that makes them very unique. It makes them very selective. Trypsin, chymotrypsin, and elastase all have different preferences. If you remember, we talked previously about the proteases trypsin, chymotrypsin, and elastase, and we said that trypsin likes to find a positively charged amino acid and cut after that. So it will find an arginine, and then it will cut the peptide bond that follows that. So the way that it works is it has what is known as a specificity pocket. In that specificity pocket, there are certain amino acids that will attract substrate amino acids. So trypsin cleaves on the C-terminal side of basic or positively charged amino acids. So arginine or lysine are the most likely targets, but sometimes histidine could be there based on the environmental pH. Chymotrypsin cleaves on the C-terminus of aromatic amino acids, and elastase cleaves on the C-terminal side of small neutral amino acids. So basically, you can think about it as the specificity pocket is what differentiates trypsin, chymotrypsin, and elastase. They're all serine proteases, so they all cut peptide bonds. They all have a serine within their active site. But one of the things that makes them different is a structural element. They either have, or they have a specificity pocket that's made up of these amino acids, or it's made up of these amino acids. And depending on the identity of those amino acids, that will impact the substrate that these enzymes are going to cut. So now back to the enzymes, or the amino acids that make up the active site of our enzyme. There's a total of 3 amino acids, a histidine, an aspartate, and a serine residue that make up what's known as a catalytic triad. So these residues and their positioning, so it's these amino acids that are central to the mechanism of serine proteases. And their positioning in the polypeptide sequence, it might be a little bit variable, but their positioning in the 3D structure of the protein, they're all going to come together and be pretty much equidistant from one another. Now, excuse me, these different amino acids are going to play different roles. Serine, for example, is an as acting as a nucleophile. Histidine will act as a general base, and aspartate will establish a hydrogen bond to orient the histidine. So you can kind of think about it as, well, all that aspartate is going to do is it's going to help position the substrate so that it can be cleaved. So chymotrypsin, what it's going to do is it's going to take a peptide and water, and it's going to release two peptides. Chymotrypsin catalyzes a rate enhancement of approximately 10 to the 9th. So when you compare the catalyzed to the uncatalyzed reaction, the mechanism is going to involve an acylation and deacylation reaction rather than direct attack by H2O. And what we see is that this reaction happens very quickly in the presence of this, it's going to happen very quickly taking nitrophenylacetate to p-nitrophenol, just to indicate our cleavage of this peptide bond. So a reaction where we looked at the conversion of p-nitrophenylacetate to the formation of p-nitrophenol, that happens quickly as opposed to our acylated enzyme. Okay, so here's our chymotrypsin mechanism. This is it broken down over the course of 7 steps. And I want you to look at this, and look at this, and realize that this is the exact same as this, though it's blown up over a couple of different slides. So first and foremost, I'd like you to walk through, or I'd like to walk you through these. So this is step number 1. Step number 2. Step number 3 is here. And then step number 4 is going to be here. Step number 5, we have the water come in. So this is 4 again. Number 6, and then number 7. Now, for the sake of simplicity and avoiding redundancy, I'm going to walk through the sequence as it's displayed here. If you like the model shown over these 2 slides, by all means, you're welcome to look at that. I like this because everything's wrapped up pretty simply. I think that, as is the case with any enzyme mechanism, it's important to identify the amino acids that are in the polypeptide of our enzyme. And that's pretty easy to do because they're all shown at the bottom of the slides, or the bottom of each one of these cells. Now I'm going to erase those, because what I want to do is show you, well, if we look at serine 195, histidine 57, and then aspartate 102, if we look at serine first, what happens with serine from cell number 1 to cell number 2? Well, immediately we see serine establishing a hydrogen bond with our substrate. Okay. In addition to that, if you look at histidine 57, histidine 57 gets mixed up into things very quickly as well. So it is, in this first cell, we have our 2 nitrogens. Each of them have a total of 3 bonds, because the one on the left-hand side has 2 carbon bonds, or nitrogen carbon bonds, and 1 nitrogen hydrogen bond. So it's got 3 bonds. The other one has a total of 3 nitrogen carbon bonds, 1 through a double bond, 2 of them through a double bond and 1 of them through a single bond. In cell number 2, we have our nitrogen now protonated. So arguably there should be a positive charge there. So where is that coming from? Well, if you look back and review, it's coming from the serine. I'm not going to dig into that too deep, because really what I want you to see is that serine and histidine change pretty quickly on in this mechanism. Now, if we look at aspartate, well, it's deprotonated. In cell number 2, it's deprotonated. Cell number 3, still deprotonated. Cell number 4 deprotonated. 5, 6, and 7. It's all deprotonated. They're all deprotonated. Now, with that said, one of the things that I want you to look back at is on this slide right here. Well, aspartate establishes a hydrogen bond, or establishes hydrogen bonds to orient or position that histidine. Okay, so aspartate 102, really all that it's doing is it's holding itself stable. It's holding itself to hold that histidine in place. Now, the good thing about that, and what I like about that, is when you inventory, okay, what all do I need to know here? Aspartate 102 doesn't do anything almost. It does actually, because it maintains a hydrogen bond, but you're never going to see that change from cell to cell. Now, with that in mind, let's go ahead and look back at that serine 195. And it's really hard to talk about serine 195 without talking about histidine 57. So I'm going to talk about both of those kind of simultaneously. So if we look at our first cell, we have serine 195 with its hydroxyl group. We have our substrate up here. Remember, our substrate is a polypeptide. So if we see this ring structure right here, that would be something like a phenylalanine. After that phenylalanine, we have our alpha carbon right here, and then we have our peptide bond right there. So that is the peptide bond that we want to break. So between this carbonyl carbon right here and this nitrogen, we want to break that bond. Okay. Now, what we're going to see going from cell 1 to cell 2 is this nitrogen on the right-hand side acting as a base. And it's going to pick up that hydrogen, which is going to allow the electrons going from that oxygen to hydrogen to establish our covalent bond with our carbonyl carbon of our substrate. So we have our covalent bond formed, which is going to ultimately be stabilized, because what we're going to see is we are going to see a positive charge emerge on the N-termini of this peptide bond, or N-termini closest to our carbonyl carbon. Excuse me. And as soon as that happens, lo and behold, our peptide bond is broken. So going from cells 2, 3, and 4, things happen pretty quickly. And when that escalates so quickly, what we now have to do is we have to get our serine and break a covalent bond. We also have to get serine and our histidine back to where they started. Now, histidine in this process actually donated its hydrogen, and it was picked up by that nitrogen to give us that positively charged group in our peptide bond that would further destabilize it. And whenever that happens, and when water comes in, now what we can have happen is the hydroxide from our water can go towards our carbonyl carbon. And when that goes towards our carbonyl carbon, well, the nitrogen on the right-hand side of our ring can pick up that hydrogen. And then what we've got is a serine, then what we have is a covalent bond between our serine and our recently cleaved peptide. We have a hydrogen that we can now lose as a proton that serine can pick up and return to its original state. And in the process, what have we done? We've broken a peptide bond after a phenylalanine. So the mechanism of this enzyme is going to be very similar to a mechanism for an enzyme like trypsin or elastase. It's going to be basically these 3 amino acids. What's going to change, on the other hand, is the makeup of our substrate. So if I saw something like, instead of this ring here, we had a nice positive charge from like an arginine or a lysine residue. Well, there we go. That's what it is. The trypsin mechanism, not the chymotrypsin mechanism. So trypsin, chymotrypsin, and elastase are all serine proteases. They all have a serine residue in their active site. Their serine residue establishes a hydrogen bond between our substrate and our enzyme. And in the midst of all that, we break our peptide bond. So this is one of the cool things about understanding enzyme mechanisms, because arguably, if you know one, you've got a pretty good grasp and understanding of others. All right, well, I hope this was helpful in helping you understand enzyme mechanisms, what different amino acids do, and basically the movement of electrons in these mechanisms. All right, well, have a good one.